Hello everyone, this is Dr. Madhusudan Rao. On the program, I teach medical students. Now I talk on the topic immunoglobulin structure and functions. I'll try to give a brief account of this so that the students will be interested to watch my video. Immunoglobulins are the antibodies. They are present in the plasma, lymph, body fluids, peritoneal fluids, CSF. They offer protection against the various infections, say viral, bacterial, or fungal. So let us know the structure of the immunoglobulin. Immunoglobulin is a glycoprotein. It has got the shape of Y. It has got a pair of heavy chains. You can see this. A pair of light chains. Because heavy chain has got the double the molecular weight when compared to the light chain. The molecular weight of the light chain is 25,000. That of the heavy chain is 50,000. So what are the properties of heavy chain and the light chain? Each heavy chain and the light chain, they have FAB segment and FC segment. What is FAB segment? FAB segment means fractured antibody segment. FC segment is crystallized fragment. FAB segment has got the variable region and the constant region. Okay, variable region means the sequence of the polypeptide in this particular region is highly variable. This variability helps the antibody to bind with the number of or innumerable antigens to which individual is exposed to. And that means the antibody binds with the antigen to which the individual is exposed to by the variable region of the FAB segment. Okay. What is constant region? The polypeptide sequence of the constant region remains the same with regard to that particular class of antibody. Otherwise, classification of the antibodies is made based upon the type of polypeptide chain at the constant region of the FC segment. Okay, so this is about the structure of the antibody. Besides, the antibody has got the region for fixing complement. So as I told you, based upon the amino acid sequence, at the FC terminal, the antibodies are classified. If the polypeptide chain is alpha, that is IgA. In IgG, it is gamma polypeptide chain. In IgM, it is mu chain. And in IgE, it is epsilon. And in IgD, it is delta chain. So based upon the polypeptide chain, at the FC terminal, the immunoglobulins are classified. So let us come into a brief account on the structure. So as I told you, immunoglobulin is also called antibody produced by immune cells, that are plasma cells, and it is a glycoprotein. It has a pair of heavy chains and light chains. Each has variable and constant regions. One unit of immunoglobulins called monomer, and the molecular weight is 1,50,000. So as you have seen in the structure, each has a pair of FAB and FC terminals are the regions. FAB is fragment antibody binding site. FC is the crystallized fragment. And you try to understand the meaning of certain terms. Epitope, paratope, and idiotope. Epitope is the antibody binding site on the antigen. So that means antibody binds with the epitope of the antigen. Right? Paratope is the antigen binding site on the antibody. Idiotope is the antibody binding site on the antibody. Right? So as you have seen in the picture, I told you it is Y-shaped, two arms and a base. FAB at the arms, FC at the base. So FAB, as we have seen, has got variable terminals and bind with the antigens. FC binds with the receptors of the immune cells. And so based upon the type of the polypeptide and the FC region, immunoglobulins are classified into, as I told you, alpha chain in IgA, gamma chain in IgG, mu chain in IgM, and delta chain in IgD, and epsilon chain in IgE. So you look at the antibody classification. In this picture, there is IgG, which is a monomer. But IgE is again a monomer because these immunoglobulins have got this single unit of immunoglobulin. That is a pair of heavy chains and a pair of light chains. IgD is also monomer. IgA is dimer. Two molecules of immunoglobulin bind by the disulfide bonds. So you got the immunoglobulin M is a pentamer bound by the disulfide bond. So this is how the classification of antibodies. So this picture shows how the antibody is binding with the antigen. The antigen has got the receptors. Okay, there are the epitopes with which antibody binds with the FAB region of the antibody. So once the antibody is bound with the antigen, it forms the antigen-antibody complex. This complex fixes the complement, right? Then the FC region gets activated. This is called the opsonization. And antigen-antibody complex with activated FC terminal binds with the receptor on the immune cells, say in the neutrophils, say the macrophages, well, followed by phagocytosis, and the antigen is destroyed, either the virus or bacteria, by the proteolytic enzymes in the cells of the immune system. That is, they could be lymphocytes, they could be macrophages, they could be neutrophils. That's how the antibody binds with the antigen, forms the antigen-antibody complex, fixes the complement in the body, and this is called the opsonization. And this activated antigen antibody complex binds with the receptors that are there, receptors that are present on the immune cells, the macrophages, etc., with the FC terminal of the immunoglobulin. This is the picture showing the antibody binding with the antigen, forming the antigen antibody complex. In certain occasions, fixes the complement, and this antigen antibody complex, the activated FC region, binds with their receptors on the immune cells, either in the circulation or in the spleen, with the macrophages. Antigen, then antibody binding site, antibody binds with the antigen, that is the epitope on the antigen. So let us go about the features of the each immunoglobulin. IgG is the important immunoglobulin, amounts to 75% of the total immunoglobulin levels. So the level of immunoglobulin G is around it is estimated around 10 to 15 milligrams per ml. This is the largest immunoglobulin circulating in the body. It is there in the intracellular and extracellular compartments, present in plasma, peritoneal fluid, lymph, and cerebrospinal fluid. It is produced as a secondary response to infection. So persists for a long time. It crosses the placental barrier from mother to infant. Whereas IgG is the immunoglobulin produced as a secondary response to infection. IgM, its serum level is 0.5 to 2 milligrams per ml. It is produced as a primary response to infection. It comprises 10% of the total immunoglobulin levels. It is intravascular in distribution, present in lymph and blood. IgA, an interesting immunoglobulin, present in the secretions as well as the mucous membranes. It provides surface immunity. It has got two types, secretory IgA and serum IgA. IgA is present in the serum in very small quantities. That is, it amounts to 0.002% of the total immunoglobulins. It is there on the surface of the mast cells and eosinophils. The FAB of the IgG binds to the allergens, carried to the receptors on the mast cells that mediate allergy, and release the mediators of allergy. So IgE is elevated in asthma, allergic rhinitis, urticaria, and other allergic conditions. IgD, its function is unknown, but it is present in the small quantities, amounting about 0.2% of the total immunoglobulins. It is present on the surface of the lymphocytes. This is a brief account of the immunoglobulins, their structure and functionality. Thank you very much for patient listening, and we'll meet you again in the next class. Until that time, bye-bye. Thank you.